Well, welcome everyone to the Ann S. Bowers Women's Brain Health Initiative Seminar Series. My name is Susan Gauthier, and I'm a neurologist that specializes in the field of multiple sclerosis. And it's my absolute pleasure to introduce our speaker today, my good friend and highly respected colleague, Dr. Amy Kuceyeski. Amy is currently a professor of mathematics and neuroscience in the radiology department at Weill Cornell Medicine and computational biology department at Cornell University. She obtained her PhD in applied mathematics from Case Western Reserve University in 2009, after which she came to Weill Cornell Medicine and never left. Thank goodness. For the past 15 years, Amy has been interested in understanding how the brain works to better diagnose, prognose, and treat neurological disease and injury. Quantitative approaches, including AI, applied to data from rapidly evolving neuroimaging techniques, have the potential to enable groundbreaking discoveries about the brain. Amy's work has focused on identifying sex differences in brain structure, function, and correlates of hormone fluctuations. Amy also has particular interest in applying computational modeling to simulation of effects of non-invasive brain stimulation and pharmacological interventions like psychedelics that may be used to modulate brain activity and promote overall brain health. I have no doubt that Amy will provide us with an outstanding talk today focused on AI and computational neuroimaging for women's brain health. Amy will speak for about 45 minutes, then we'll have 15 minutes of Q&A. Participants can put their questions in the QA box at any time. So welcome, Amy.
Thank you so much, Susan. So yeah, it's my pleasure to be here this morning to tell you all about some progress that we've been making in our lab using computational approaches to analyze neuroimaging data and applied to sex differences and looking at women's brain health. So I'm the co-director of the AI core for the Ann S. Bowers Women's Brain Health Initiative, along with Nina Milan, my colleague there. So today I want to start with, let me make sure that this is, okay, now you can see my screen without the Zoom. All right, so as you probably all know, March is Women's History Month. So I thought I'd start us off with some really interesting examples of women throughout history who have been working in computer science. So women were actually the first computers. So that's the technical term that was used to describe what they did. Which was running the mechanical part of the computing machines a long time ago. But even before that, so in 1830, we have Ada Lovelace, who was the inventor of the algorithm. So she was actually described as a poetical scientist, which I guess is not surprising since she was the daughter of Lord Byron. But she was the first to realize that computing machines could be used for more than just computing. And this was even before computers were even made, or they were conceived of, the Charles Babbage compute machine. But it was even before the computers or computing machines were even built. And next we have Edith Clark. She was a pioneering electrical engineer at the turn of the 20th century, and she was a computer, quote unquote, and she actually became the first female electrical engineer in America in 1922. So she was at the leading edge of that. And I just recently learned about this. I'd not heard of this before. This is called the ENIAC programmers. There were five women: Betty Snyder, Kathleen McNulty, Jean Jennings Bartik, Ruth Lichterman, and Frances Bilas and Marilyn Westcoff. And they were tasked with figuring out how to program the world's first general purpose programmable all-electric computer, which was known as the ENIAC. And this was done during World War II. And this was without manuals or programming languages because none existed. So this was an effort that was happening during World War II, and it was kept under wraps until shortly after, when it was released, the information about them was released in 1946. And then, of course, fast forwarding to the Cold War, Katherine Johnson was an African-American space scientist and mathematician, and she was instrumental in calculating key flight trajectories for the space race. She also worked as an advocate for women in STEM throughout her life. And then many of you may know Grace Hopper. She was a famous computer scientist working in language development, and her famous language COBOL is still in use today. It was the first language to be written in English rather than computer notation, and she also was a very strong advocate for more women in STEM. And of course, this is only just a few of the many that exist throughout computer science and electrical engineering, so many I can't name. But I do want to point out that while there are these strong female characters throughout history of computer science and electrical engineering, unfortunately, there's a gender imbalance to this day in engineering and computer science generally. So here we have along the x-axis time, starting in 1870 and ending about 2015. And the y-axis is the proportion of women in each of these areas. So you can see things like health professions and education and foreign languages are more female centric, whereas things like engineering and computer science are sort of at the bottom of the pile. So they're about 20%, which is in my experience, the general ratio of male to females in engineering and computer science departments across the board. And there's this interesting trend where until 1950, there was a rise in women in computer science. And after 1985 or so, there was a strong decrease. There's lots of theories about why that might happen, including sort of the advent of the personal computer and things like that. But I don't think it's really conclusive to this day. So we need to we have some work to do. All right, let's turn our attention to the brain, which is what we're all here to talk about. And I'll start off with my sort of favorite poem. This is by Emily Dickinson. And the first stanza there is, "The brain is wider than the sky, for put them side by side, and one the other will contain with ease and you beside." So it's this beautiful sort of quasi-scientific philosophical view of the brain, which is to our knowledge, the most complex object in the known universe. A typical human brain has about 86 billion neurons. For reference, there are an estimated 100 billion stars in the Milky Way galaxy. And in addition, between those 86 billion neurons, there are 100 trillion connections. So it's this very complex and complicated object. And our understanding of it is we're just scratching the surface of it. But a convergence of 3 key advances in the past 5 to 10 years has really enabled unprecedented discoveries about the brain's structure and function. By the way, anytime you see this little square here, I used DALL-E 2 to generate some of these images. And I always had to put woman scientist because otherwise it was a man. So I'll talk a little bit more about bias in data in a minute or two. So the first sort of convergence of these key developments is the emergence of publicly available big neuroscientific data. So these are kind of released under these collection and sharing protocols like the Human Connectome Project, the Enigma and brain initiatives. And these contain neuroimaging techniques, data from neuroimaging techniques that allow in vivo measures of the human brain, either activity or anatomy, and also usually some sort of measure of behavior or cognition. And this allows us to do what we call brain behavior mapping, which is relating the measures that we extract from neuroimaging to some kind of external variable or behavior. And I'll talk a little bit about that in a minute. The second key advance is deep learning's reemergence. So the perceptron was actually first conceived in the 50s and late 50s, early 60s. One of the people that did this was Frank Rosenblatt at Cornell, and it was kind of ahead of its time. So there was an AI winter for quite some time, which was apparently driven by social pressures and a lack of compute power. And it was only several years ago that deep learning started to take over most of modern machine learning applications, from computer vision to large language models, which I'm sure you've all interacted with at some point in the past year. And then finally, of course, it's the advent of sort of GPUs whose structure is optimal for deep neural network architectures. And so these 3 key advances have kind of paved the way for these unprecedented discoveries of how the brain works. So how are we using these discoveries? So I see generally 2 broad categories of questions about the brain that AI is helping us try to answer. One is more basic, how the brain works, and one is more clinical, how can we fix it? So first, how does the brain work? So there's lots of exciting experiments being done involving recording an entire brain's worth of neurons, which is like calcium imaging in mice, not in humans, of course, while the animal is undergoing a learning task. So in these studies, you can actually record an entire brain's worth of neurons during a learning. So we can really get this large amount of data to understand how a mouse is actually learning. But in terms of human studies, these huge sets of neuroimaging data across various populations is allowing us to map these various neuroimaging biomarkers to things like age, sex, cognitive performance, and environmental variables to see how intrinsic and extrinsic factors can change brain anatomy and physiology. We also have a lot of studies looking at how the brain responds to incoming signals from the environment. So encoding models, that's looking at how brain activity responds to things that the person is looking at. And so understanding this mapping might allow us to understand how this very complex system integrates information from the environment and translates that into action. And it's sort of the first step in mapping how humans interpret and respond appropriately to their environment. And this also has implications for building AI because we can use inference from how the biological brain works to improve artificial neural networks as well. So there's a whole host of research activity going on in comparing how brains and artificial neural networks align, and actually improving artificial neural networks using biological principles. And then another area of major inquiry, these large data sets has allowed us to to investigate what makes individuals unique. So this so-called fingerprinting of the brain. And this potentially has implications for personalized medicine and predicting propensity for certain mental health disorders, for example. Okay, and so in terms of women's brain health, these are just a few areas of research or questions related to women's brain health in each of these areas. So on the brain behavior map side, we have how hormones may impact the brain, looking at sex-specific development, how oral contraceptives may change brain anatomy and physiology, as well as menstrual cycle. I'm calling them brain event maps, so things like pregnancy, motherhood, and menopause, how these things can change the brain, as well as looking at genetic influences in brain structure and function. And then another area, this sort of encoding and decoding, is looking at sex differences in visual and auditory processing. There's some really interesting preliminary work looking at visual and auditory processing and how it fluctuates across the menstrual cycle. So that was pretty interesting as well, but I think it's a very early area of research and one that's ripe for more work. And then also looking at individual variability in brain structure and function or activation across all of these different events and sort of behavioral outcomes. And this is something that Nina, I know, is very interested in, is digital twins. So it's sort of making an in silico version of a person so that you can see what happens if you perhaps give them a certain drug or give them some certain treatment. And you can sort of see that particular person's response to that treatment or intervention. Then the next question AI can help us answer is, how can we fix the brain? And so AI has a lot of applications to neuroimaging data collected in the clinic, including image analysis, so things like volumetrics and segmentation of brain structures and automated anomaly detection of things like tumors or lesions, and of course, image reconstruction improvement. So using AI to sort of make better images that come off an MRI scanner, for example. And then once we have this set of brain imaging based biomarkers, we can use machine learning or AI models to map them to disease or some other continuous measure of impairment or symptomology. And these models can help us diagnose individuals with diseases or disorders, especially ones that are difficult to diagnose or may benefit from early detection. So things like Alzheimer's, autism, schizophrenia, OCD, or early childhood developmental disorders that are better to be identified earlier. And we can also use this pathological signature information to develop accurate prognoses. So it's important for patients and their families, obviously, when planning care, particularly in areas where clinicians have difficulty in predicting how someone will recover. So for example, brain injury. And AI applied to neuroimaging can also help us in personalized treatment selection. So again, this goes back to that sort of digital twin idea. For example, AI can help us identify subgroups of subjects that might be more likely to benefit from a certain therapy, for example, TMS in treatment-resistant depression. And finally, these pathological signatures, this is really important, can point us toward possible mechanisms of disease and toward areas that we may be able to intervene to alleviate symptoms. So things like lesion symptom mapping or autism even. And then finally, not something I'm going to be talking too much about today, but this is a really popular area of research right now. This is brain-machine interfaces. So AI can be actually used to read neurons' commands and relay these commands to a robotic arm, for example, or an exoskeleton. To aid people who are paralyzed, or even to allow people to communicate when they can't otherwise. So things like ALS or locked-in syndrome, people can, they have this training session with patterns of brain activity that map to certain words, so that this AI can sort of translate their neural activity to language. So again, I want to highlight some potential areas of women's health-related research questions in each of these areas. First, which is something important that I think we should investigate that hasn't been so far, which is, do these AI-based volumetric and segmentations work equally well across sexes? So I actually think that's not something that's been done to my knowledge. And that's very important if we're going to start using these automatic segmentation and volumetric analyses for sort of clinical applications. And then, of course, using these models of diagnosis, prognosis, and treatment in female-only disorders, things like PMDD or postpartum depression, perhaps identifying if somebody is at a higher risk for postpartum depression so that they can intervene earlier. And then, of course, looking at things like sex-specific differences in the likelihood of disorders. So why do women get MS more often than men? Why does ADHD symptomology sort of vary between the sexes as well? And also looking at sex-specific differences in the pathology or treatment response. So, of course, this one example is in Alzheimer's, where there was a treatment that was approved, and it looks to be more effective in males than it is in females, and we want to know why. So we may be able to use AI or machine learning models to tell us these answers. And then, of course, something else that has not been looked at either in brain-machine interfaces is, are there sex or hormone-specific drift in neural representations, which I think is really important if we're going to be using these things to help people communicate who can't otherwise communicate, or to move a robotic arm for paralyzed individuals. Okay, so let's move on. This is a review that my former PhD student, Elvisha Damala, and I have been working on. And it's looking at sort of sex differences in the brain and disorders of the brain. So here we have these sort of stacked bar plots of the prevalence of the cases per 100,000 from females on the left and males on the right. So you can kind of look through that. And on the y-axis is the age of onset of these different diseases and disorders. So on the left, we have neurodevelopmental disorders, neuropsychiatric disorders, and neurodegenerative disorders. So you can kind of take a look at that while I kind of tell you a little bit more about our motivation for writing this paper. So a lot of neuroimaging studies have identified sex differences in the brain, and most reliable are volumetric differences. Males have larger global cortical gray matter, white matter, subcortical gray matter, and ventricular volumes, while females have stronger white matter connections between hemispheres, and males have stronger within hemisphere connectivity. But the opposite is true in the cerebellum, actually, which is pretty interesting. Females exhibit greater within network functional connectivity, and males have greater between network functional connectivity. I'll talk a little bit more about functional connectivity in a minute. And these differences likely arise from a complex interplay of biological elements like genetics, hormones, and development, and also sociocultural factors. There are also sex differences in brain disorders. And so again, this what the stacked bar plot is showing. So in addition to sex differences in prevalence, there are also sex differences in symptom presentation. So we know that, for example, things like in ADHD, which is often underdiagnosed in females because they have more inattentive symptoms compared to boys who have relatively higher rates of hyperactivity, which is easier to detect if you're a parent. There are also sex differences in the neurobiological correlates of various brain disorders. So example, again, in ADHD, the strength of the association of ADHD traits and basal ganglia volume, for example, varied between boys and girls. And that was in an ABCD study. It's a big cohort of 9 and 10-year-olds. Okay, so to get to some of our results, so this is the talk outline. I'm going to start with an example of brain behavior mapping, and then go through 3 examples of brain structure mapping, and sort of how that is impacted by sex and different female hormones. Okay, so first, just to get us all on the same page, because I'm not sure the background of everyone, I'm going to be talking exclusively about MRI. So MRI, this is an MRI machine here in the middle, and it's a big magnet. And what it can allow us to do is take a very detailed picture of the brain's anatomy. So that's an example at the top there. That's an image of the brain's anatomy. You can see the ventricles and the sagittal plane here, where the nose and the eyes are. And then on the left, I have an example of diffusion MRI. So diffusion MRI is a way of tracking the diffusion of water molecules in the brain. And on these sort of wiring between different parts of the brain, the white matter, it's very thin. You can imagine it like electrical wiring. And as water diffuses, it goes along the long axis of those wires more than it goes across the short axis. So you can look at the shape of the diffusion in each voxel and reconstruct these likely white matter pathways that connect different parts of the brain. So this is a way to determine what we call structural connectivity. And then on the right, we have an example of the functional connectivity, or the functional networks in the brain. And this is measured by using blood oxygenation level dependent signal, or the BOLD signal. Many of you might know it as functional MRI. And so this is actually tracking the oxygenation of the blood in the brain. And so when a neuron fires, presumably, and studies have shown that this is relatively true, as a certain area of the brain starts to activate, blood is rerouted to divert resources and metabolic precursors to those areas. And so you can track the regions of the brain that are active at any given point in time. And we can take a picture of the brain just about every 1 second or so. And so if you have these sort of two measures of structural connectivity and functional connectivity, or actually, so I should preface the connectome by saying that to calculate this sort of network at the bottom, so that here we have an example of a matrix, we chop up the brain into parts. So we say, you know, this is the left DLFPC, this is the right DLFPC, this is the thalamus. So we have these different parcels in the brain. And then what we can do from the diffusion MRI is sort of count the number of white matter tracks that are connecting pairs of regions. And so once we do that for every pair of regions, we can get a matrix that tells us the strength of each pair of regions' structural connectivity. So the higher the value there, the more the white matter is that connects those two regions. On the flip side, we have the functional imaging, and there we can take every voxel that's in each of these regions of the brain and average them to get a time series for that region. And then we can take each region pair's time series and just correlate them, let's say, and see how similar they are. So if they're very similar, that means their time courses are very similar. They're firing together, they're depolarizing and polarizing together. And so there we can correlate those and get a measure of how functionally related they are. So those are the two sort of connectomes, structural connectome and functional connectome. And so once we have these measures extracted, we do the MRI, we extract the biomarkers. To perform brain behavior mapping, what we want to do is then take some cognitive, physical, or behavioral test, let's say, from outside of the scanner. And then what we do is we create some statistical or machine learning or AI models that allow us to map between the biomarkers and the behavioral tests. So that we can then map the relationship between brain structure and function to some sort of external variable of cognitive or behavioral function. So I'm going to run through quickly an example of brain behavior mapping. And this was work that was done by my former PhD student, who's now a faculty member. And here what we wanted to look at was, do men and women, or males and females, have different relationships between the brain's functional connectome and different cognitive outcomes? And so this was motivated by some previous work that was using sex-specific models, and there they suggested that the female IQ is more predictable than male IQ using functional connectivity. So here you can see a scatter plot of predicted and true values for females, and the correlation is about 0.7, and for males it was about 0.46. So the idea was that perhaps the female IQ was more predictable from functional connectivity than males. They also investigated the coefficients in the model for the brain behavior map, which is one way that you can sort of identify connections or regions that are important for predicting some outcome. And so here they have the male and female coefficients, and you can see they're very different. And on the right, you can see there were only sort of 3 connections, which are listed in black, that were shared between the sexes. And so it seems like from this previous work that there could be potentially differences in the mapping between brain behavior across the sexes. And so what we did was we used the HCP data. This is the Healthy Human Connectome Project, young adult data. So this is a collection of about 1,200 subjects between the ages of 26 and 35. And there we looked at the unrelated set of subjects. This is about 420 healthy adults. And we trained models to predict different cognitive outcomes. So on the y-axis here, or x-axis, you can see the different cognitive scores. And on the y-axis, you can see the model's prediction accuracy for 4 different kinds. So we trained the model just on male data and tested it just on male data. Same for females in orange. And then we did the opposite for the other 2. We trained on female data and tested on male data, trained on male data and tested on female data. And this was to analyze if something that we learn about the brain behavior mapping from females could be applied to males and vice versa. And we actually see that most of the models do perform similarly well when they're trained and tested on each of the different sexes. There were a few examples where the male trained and male tested models did better than the others. And so that was in fluid composite and this dimensional change card sort. But for the most part, they were pretty similar. And then we looked at the sort of functional connections that were important in predicting these various cognitive outcomes. So here's an example of crystal and fluid abilities and the mapping for the functional connections between them. And largely we see that feature importances are shared between males and females, indicating that there's not much difference in the brain behavior relationships across sexes. So these are correlations of the model's coefficients for the male and female tested models in the upper right-hand corner and female-male models in the left-hand corner. So these are revealing that there are very similar mappings between functional connectivity and cognition between the sexes. So I've been working with Qingyu Zhao. He's been spearheading this really cool project, a perspective paper about future considerations for machine learning-based brain behavior models. So of course, there's heterogeneity in all of these at all of these levels. So it's heterogeneity in the brain. So there's lots of studies looking at sort of doing unsupervised clustering, and you can identify these subtypes of neural activation or structure. So there's definitely differences in people's brains, and they might be sort of subgroups. That's what's being highlighted here. There are these subgroups that are more similar to each other than they are to the other groups. And then on the other side, there's phenotype heterogeneity. So there's definitely, cadres of symptoms that you can have in let's say, depression. There's cadres of symptoms that you have, and people have done sort of unsupervised clustering to identify these subgroups of people with more similar phenotypes than others. But then what's been ignored so far, I think, for the most part, is this relationship level heterogeneity. So looking at how allowing the mapping between brain and behavior to vary. So let's say looking at mapping subtype 3 over here in the neural domain to, let's say, subtype 1 over here in the phenotype heterogeneity domain. And one way that we thought about doing this is using, let's say, for example, a nonlinear machine learning model here represented in this bottom sort of left-hand corner. The heterogeneity of this relationship may be modeled as a second hypernetwork that might act as a generator for the parameters of the original network, mapping brain to phenotype. And this generator would then allow us to model external variables like sex, age, and environment, and how they might impact the neural network itself. And so, for example, in women's brain health, we could think about how sex or hormones or menopausal stage may impact this mapping from brain to behavior. And then another way we thought about doing this could be through a mixture of experts modeling approach, where the final mapping is sort of a combination of these several categories of expert models trained for various neural subtypes, where the combination is determined by these external factors. So this is one thing that we're working on currently. Okay, so next I want to look at brain structure function modeling. So again, I told you a little bit about diffusion MRI and functional MRI and how we extract structural connectivity and functional connectivity. And you can imagine that it would be sort of straightforward to imagine to map structure to function, right? So you can imagine that if two regions have wiring between them, you can probably think, oh, they're probably functionally related too, because when one fires, the other one fires. But it turns out that's not true. So if you just take the structural connectome and the functional connectome and you correlate them, you get about 0.25 or 0.3. So it's not very related. And that's probably good, because that allows us to have a very complex functional dynamics. And so one of the ways that a lot of people have been looking at mapping the structure and function and using models to map the structure to function, including things like neural mass models. So in the upper left-hand corner here, this is an example from the Virtual Brain Project, which are each region in the brain has its own system of differential equations, and then you sort of link them using the structural connectome. And there's also sort of statistical approaches using just regression on the structural connectome or metrics extracted from the structural connectome to predict the functional connectome. On the left here, we have graph theoretical models, so things like looking at the topology of the system and mapping the topologies of the two networks to each other. And then, of course, recently we've been looking at machine learning and sort of trying to, in a data-driven way, map the structure to function. And finally, we've been looking at an engineering approach called network control theory, which I'll talk a little bit more about in a minute. But there have been lots of studies that have focused on investigating the changes in structure-function relationship in disease or recovery from injury, including neurological and psychiatric disorders. And here we have sort of two studies that are highlighted using the virtual brain network or the virtual brain framework, which is, again, this linked neural mass models. And those two studies actually showed a correlation between the model parameters that they fitted to the data and cognition collected in Alzheimer's patients and motor impairment after stroke. TBI, traumatic brain injury, is another example where a recent study showed damage to the structural connectome causes decreases in the metastability of the brain's activity patterns, which were also correlated with worse cognitive outcomes. And then finally, these really cool studies have been showing in depression and disorders of consciousness have been showing that a stronger structure function coupling, so the collapse of the functional connectome to the structural connectome, was actually related to worse depression scores and more severe disorders of consciousness. So looking at the structure function relationship is very important. And one paper, this is a work from my former PhD student, Zijin Gu, where we looked at mapping structure and function in just a merely statistical way. So here again, we have an example of the structural connectome and the functional connectome. And all we did here to create a structure function coupling measure for each region in the brain was to just take the row of the structural connectome, so that region's connectivity profile to the rest of the brain, and the same region's row in the functional connectivity connectome, so that region's functional connectivity to everywhere else in the brain, to get a strength of how aligned the structure and function was. So here's a region plot of that. So the yellow are more structural function aligned. So things like visual and subcortical regions had the strongest coupling, and it was weakest in the limbic and default mode regions. It was consistent across time and in different sample populations. So looking at this coupling, we thought we would see how the coupling was related to things like age, sex, and cognition, and to also investigate if it was heritable. And so we generally saw that there was an association with age in a few regions, and it was generally, this coupling was stronger in males than it was in females. So you can see these regions that are outlined in black as the significant regions. And then we also found that the weaker coupling or stronger coupling in the right insula and weaker coupling in bilateral middle cingulate were related to higher total cognition scores. So it seems like there might be some relationships with some of these regions coupling and cognition. And we also showed that it was heritable. And it was not heritable in a way that was reflective of just function or structure. It was actually heritable in a different pattern of way. So this was an example of how we can map neuroimaging biomarkers of structure and function to individual characteristics using computational approaches. And next, I want to talk a bit about machine learning. So again, we talked a little, I mentioned this in the first slide, looking at using artificial neural networks or deep learning to map between the flavors of the structural and functional connectivity to try and understand the relationship between structure and function. So this is work that is being spearheaded by my PhD student, Keith Jamieson, who has been sort of creating this really massive autoencoder framework. We call it the crack encoder because of all the arms. So it's one of the best names I think we've ever come up with. But it allows us to map between flavors of structural connectivity and functional connectivity. So here you can see there's lots of different ways to extract. So if people have worked with this data before, you know there's lots of choices that go into how you process the data, right? So there's, do you do global signal regression? Do you do high pass filtering? Do you do band pass filtering? Do you use a different atlas? So there's all these sort of choices, and they can impact the final brain behavior map that you find. And so what we wanted to do was find a way to sort of mush these together. So for lack of a better word. So we have all of these different flavors of structural and functional connectivity, and we created an autoencoder for each one of those, and an encoder and a decoder, and we pushed them all into the same shared latent space. So it's sort of a way of doing dimensionality reduction of multiple connectome flavors, but pushing them into a shared common space. And so here, I'm not going to go through each of these because it's a little complex, but all I want to say is that the secret sauce to this model is that every single person has about 15 functional and structural mappings. And what Keith did there was he required that the model train for the latent space representation of each of these connectome flavors to be very similar for the same subject and dissimilar from everyone else. So it's something called contrastive learning. So you try to push the same subjects connectome flavors together and apart from everyone else. And so one reason we wanted to do that was because one thing that we've been noticing in the literature is that when people do these sort of models to map between brain and behavior, or between structure and function, they look at the correlation between the predicted functional connectivity and the true functional connectivity, for example. However, if you just take the mean functional connectivity and correlate it with the true functional connectivity, you get a pretty high value. So that's what's here on the right. So if you look at the correlation between the true functional connectivity of a person and the mean, the average correlation is about 0.95. So it's very high. So what we wanted to do is create a metric that allows us to see how identifiable our predictions are. So that's something called fingerprinting, if people are sort of know about this thing. But it's kind of highlighted on the left here. So here we have the true and the predicted, and we correlate everybody in our test set. So let's say we have about 80, 84, 85 people in our test set, and we correlate everyone's true and everyone's predicted. And hopefully what we get is a maximum along the diagonal, because that shows that our true and predicted are matching best to ourselves. And so here, it's just an example of how we can actually calculate this top one accuracy. So we call it top one accuracy. So where am I in the, if I correlate all the predicted and true, is myself my own true connectivity in the top one? If it's not, maybe it's in the second. So we could actually calculate the average rank percentile. So that basically tells us if we're not in the top, how close are we generally? So those are the two metrics I'm going to talk about, which is top one accuracy and average rank percentile. Okay, so here we have all of these sort of flavors of functional connectivity on the y-axis, and all of the other the same flavors of structure and functional connectivity on the x-axis, and we're mapping between flavors. So every pair of flavors has a top one accuracy, and you can see we do really well in mapping between flavors of functional connectivity, and we do pretty well mapping between flavors of structural connectivity. And between structure and function, of course, there's a drop, but we still do actually pretty well. We have about 52 to 54% top one accuracy. So that means about half of the subjects are being matched to themselves with their predictions. And just for comparison, using related subjects as the prediction, when you use population mean, you don't get any top one accuracy. When you use unrelated people to match to other unrelated people, you don't get very high accuracy. And you start to get the similar accuracy to monozygotic sort of or dizygotic twins, which is what we get in that crack encoder, which is about 54%. And we compared it to a couple of state-of-the-art sort of structure to function mappings, and the top one accuracy is about 2 to 3%. So we're doing much better than that. Random is 1% basically. And if we look at the average rank percentile, so if I'm not in the top one, am I still pretty high up? And the average rank percentile is about 97 and 98, which means we're generally right at the top 2 to 3%, even if we're not at the top. And the average rank percentile for the current state of the art is, again, a round random chance, which is 53 to 50%. And the current state of the art is 53 to 55%. So we're doing a pretty good job of preserving individual differences in the mapping between these flavors of connectivity. One thing that the crack encoder allows us to do also is to fuse multimodal connectome information. So one of the motivations of doing this was because there are all these choices in the pipeline, usually what we do is we replicate our findings on all of these different choices, right? But wouldn't it be interesting to take these all as snapshots of the brain and try to use them to average out and get some better representation in this latent space, perhaps? And so what it allows us to do then is to take each of these flavors of connectivity, map them into this common space, and then take the average. So we have this sort of single representation of all of these different flavors of the snapshots of the brain in this latent space. And we still see that latent space, after we decode and do the identifiability, it has even higher identifiability for some of these sort of mappings. So that latent space then, the next step was to figure out if that latent space was actually behaviorally or demographically relevant, or if it's heritable even. So one of the ways we validated that latent space and the information contained in the latent space was we looked at the relatedness of subjects. So here we have the unrelated siblings, dizygotic twins, and monozygotic and test-retest, which is self. And you can see that the similarity in the latent space increases with increasing relatedness. And then one really interesting thing we found relevant to our discussion here is that when you project the latent space into two principal components and you plot by sex, red is female and male is blue, you get a pretty good separation. And so actually, if you use the latent space to do demographic and cognitive predictions, brain behavior mappings, we actually see that the latent space does just as well or better than the raw structure function connectivity, particularly better than the raw structure function connectivity in the cognitive predictions. So this latent space is allowing us to perhaps combine information from different sources into a common space and also isolate brain behavior mapping relationships. We then, I'm not going to go into that too much because I see I'm running out of time, but we did apply it to the development and aging data sets, and we see we get pretty good identifiability. This is even without any retraining at all. It was just using the straight crack encoder trained on the young adult data. And it does pretty well for the developing and aging populations as well. Okay, so finally, I want to get to the network control theory. So we know that the brain is this complex network system, but it can be controlled. So we know that we can respond to things in our environment appropriately and complete everyday tasks using what we call sort of cognitive control, or control of your brain's function. So there's actually an engineering analogy to cognitive control, wherein the state of a complex system can be modulated by some external or internal energetic input. And this energetic input allows the system to achieve different states by traveling along these trajectories in the dynamic landscape. So you can see here, this is state 0, and you can move along this trajectory to a different state. So the brain is actually a really interesting example of a complex system with a defined pattern of interconnectedness. So in this case, it's the structural connectome. And it can be represented as a network with nodes, which are gray matter regions, and edges, the white matter connections. And then the pattern of brain activity at any given point in time is the brain state. So here's just an example, sort of visually, of that. So here we have time from our fMRI data, and we have regions in the brain, and we sort of put them all into a big pile and cluster them, for example, using data-driven approaches like k-means, to identify commonly recurring states of brain activation. And then you can map those back to the original time series, and for each person in each scan, you can get like a sequence of states. So how the brain moves through these states in its functional space. And then in network control theory, the brain is considered this linear system where the activity which is x at time t, flows passively through the network via diffusion process on the structural connectome A, and also actively through the effect of external inputs that drive the system. So these are the inputs that could be driving the system to one state or another. So just an example of that, we have this, let's say we're at this initial state x naught, and we want to get to this final state xf. If we have an uncontrolled diffusion process, it might not take us in the direction that we want. But if we have this sort of control energy that's input into the system, we can drive it to a state that we want to get it to. So there's some really interesting examples, and one of the first is from Danny Bassett's lab. So it's in 2020, looking at fractional occupancy of states and how state transition probabilities shifted from rest to task. So here you can see in the NBAC task, fractional occupancy of these VIS networks, visual networks, were higher, and default mode network occupancy was lower. And then they also mapped sort of probabilities of transitioning between different states, and they actually correlated with worse and better working memory performance. And then they also found that whole brain sort of control energy from any state to the default mode network state was decreased with increasing age. So it seems like this network control theory approach can be used to investigate the brain, and we've done so quite extensively over the past couple of years. Parker Singleton has been looking at how the energy changes in psychedelics and heavy alcohol use. Jaron Toslu has been looking at changes in the transition energy landscape in MS. And Nate Roy has been looking at transition energy landscapes in mild TBI throughout recovery. And so what we actually applied this to is this Emily Jacobs and Laura Pritchett's data from the 28andMe data set. And we actually found, so this was data that's collected, for those of you that don't know, one scan every day over 30 days from one single female. And they had fMRI and hormone levels. That's what's relevant for our study here. And what we found was a significant partial correlation, was a pretty a moderate correlation between estradiol levels and global transition energy. So what that means is that the days where estradiol was higher, the transition energy required to complete the state transitions was actually higher as well. And so then we investigated region level transition energies related to estradiol. And we actually found that the right thalamus, left inferior temporal, and bilateral precuneus had significant relationships with estradiol after considering frame-wise displacement or motion in the scanner. And it's interesting because precuneus functions include recollection and memory, integration of information relating to perception of the environment, cue reactivity, mental imagery strategies, and episodic memory retrieval, and effective responses to pain. So I thought this was a really interesting finding. And we also saw widespread increases in the state pair transition energy. So we have all these states, and you have transitions between every pair, and you correlate the transition energy of each of these transitions with estradiol. They were particularly high for a state that had low amplitude activity in the frontal parietal and visual networks, and high activity in the limbic network. We also found, interestingly, in the HCP aging data set, so this is 347 women between the ages of 36 and 100 with MRI and hormones, as well as other demographic and behavioral information. And we actually found the same relationship as in the 28andMe data set, where increased estrogen and decreased entropy, I didn't talk about entropy before, but that's also something that goes hand in hand with transition energy. And they were associated with increased estradiol. But we know that age has an impact on hormones and transition energy from previous knowledge. So we performed a causal mediation analysis to disentangle this complex relationship between age, hormone levels, and a brain metric of global transition energy. And we actually found that most of the association between age and transition energy was mediated by estradiol. So that is the relationship between age and this transition energy are mostly accounted for by the impact of hormones that are changing with age. We also found, interestingly, that this increase in transition energy at a global level was mostly driven by the increased transition energy related to estradiol in and out of the frontal parietal state, which I thought this was pretty interesting. And it's characterized by this high amplitude activity in the FPN. So you can see a correlation here between the frontal parietal state transition energy and estradiol. And then finally, at a regional level versus estradiol, we identified 3 regions with significant relationships with estradiol and transition energy after accounting for age and motion. And this included the bilateral postcentral gyrus and left paracentral. And this is interesting because these are primary somatosensory cortex. And it's been shown in previous studies that somatosensation can vary across menstrual cycles. So there's higher sensitivity during high estrogen phases. So I thought this was a pretty interesting finding. Moving on to the ABCD data set. So this was work that's been done by my PhD student, Louisa Schilling. And here we looked at the ABCD data, which is, if people don't know, the first release of this ABCD data was thousands of individuals who are 9 to 10 years old, males and females both, with a resting state fMRI, diffusion MRI, and also behavioral metrics and diagnoses. And so what we did there was we applied this network control theory framework, and we actually found that females have larger global transition energy than males, and it was driven by sort of default mode network, ventral attention, and somatomotor networks. So there's this drastic sort of increase in females, but this is for ABCD, so this is 9 to 10 years of age. If you extend it out in the HCPD data set, which is individuals between 8 and 22 years of age, you actually see something very interesting. So at the 9 and 10, we have a replication of our findings that females are greater than males. But as they go through development, males increase at a more rapid rate than females, and they cross at about 14 years of age and become the opposite, where males have greater transition energy than females. So it seems that there are different trajectories throughout development of these metrics of structure and function relationships. We also, Louisa and I were very interested in looking at family history of substance use disorder. And so here we have an example of females are on the left and males are on the right. This sort of red color is as family history negative. So it's people who don't have a family history. And the blue are family history positive, or FHP. So they do have a family history. And you can actually see that there's a sex-dependent effect of substance use disorder in males and females, where females with a family history have higher global transition energies than females without a family history. And the opposite is true for males. So we might even have opposite findings across sexes in some of these metrics. When we look at the transition energy at a network level, we see that somatomotor networks actually had higher transition energy in individuals of the family of substance use disorder, regardless of sex. So that's at the top. It says both males and females together. And we also see this trend reflected at the global level. At the network level, females with a family history of substance use had higher transition energies than those with no family history. And it was particularly strong in the default mode network and somatomotor. And then again, on the right, we had the opposite, where males with a family history had lower transition energy in the dorsal attention and ventral attention and higher somatomotor energy compared to males without. And this was actually a sex-specific correlation was found with density of family history. So not just looking at the two groups, but also counting how many people in the family had substance use disorder. And the sort of correlation with only females in the default mode network was significant and not significant for males, and the opposite was true for the dorsal attention network. Finally, I'd like to talk about this work that I've been doing with Marie Hedo, who's an incoming PhD student of mine, looking at ADHD and sex-specific sort of neural signatures of ADHD. And here we again looked at the ABCD data. And on the left here, we have just a group analysis of ADHD versus typical development, but separated out by sex. And you can actually see that the limbic transition energy, limbic network transition energy was higher in girls with ADHD, but not for boys with ADHD when compared to typically developing individuals of the same sex. And the transition energy in several subcortical and frontotemporal regions had higher TE in girls with ADHD compared to typically developing girls. And that included left amygdala and bilateral nucleus accumbens, which is really interesting because it's dopamine enriched, but none were significant for boys. If we also look at correlations with ADHD symptom severity, it was significant for limbic and frontal parietal networks were associated with higher TE. But the relationship between limbic TE and ADHD severity was varied by sex. So it was positive for females and not for males. So we're again seeing this replication of the idea that the ADHD symptomology and neural signatures vary by sex. All right, so let me wrap up. I see I only have a few minutes left, so I want to be able to get questions. So I'll go through this quickly. So again, data right now is Nina and I's biggest problem. So there's a lot of publicly available data sets, and they're invaluable resources, and I love them all, but not all of them are fair, and not all of them contain the markers that we want. So of course, bias can creep in. This is one of the things that we need to highlight when we're working with AI is that biases that exist in the data will be reflected by the AI algorithm. And if you aren't aware of that, then the AI output can appear objective when it's not. So a recent analysis of NIH-funded human neuroimaging databases revealed that a large portion of those studied were white and non-Hispanic origin. So using these data sets without some sort of clever approaches, which could be possible, that may mean that the conclusions or inferences are biased towards the main group. We also have a problem. So these are a big example of a lot of the data sets that exist out there that are publicly available. And I've highlighted a few that have some female metrics. So things like oral contraceptive history, reproductive history, hormone levels, menopausal staging, menopausal severity, premenstrual dysmorphic disorders severity. So a lot of these things are not being collected. And if they were, we would be able to do these bigger analyses of sort of using AI approaches applied to these big data sets. Okay, so yeah, in conclusion, we can use these computational models and AI and with these big data sets that contain women's brain health metrics to look at how the brain works and how we can fix it, and potentially have translational applications in clinical practice. But again, the AI needs are that we need more data and more data that contains women's health information, which is where Emily is coming in and the WBHI is coming in to sort of swoop in and save us. And we also need explainable and personalized models. So that's something we consider when we're using AI applied to clinical imaging. And then, of course, we need more AI and computational researchers that are interested in women's brain health. So to that end, you can join us. I have 2 postdoc positions available right now, working with the WBHI. And I think we're a pretty good group. So come and join us. With that, I'll stop and we can do some questions.
Wow, that was awesome. Thank you.